You drank your protein shake after the gym? Sure, it goes straight to your muscles. Actually, it doesn't. Chicken protein can't just move from your plate into your muscle. First, the body takes it apart into pieces, and only then decides where to send them. And the muscles don't get all of it? That is even trickier. It doesn't dissolve in water, yet it still reaches the blood by a detour almost nobody has heard of. So how does food even cross the wall of the intestine? And why does that wall let the good stuff through, but hold back bacteria? Let's start with protein, and with that very gym myth. Picture this. You eat a piece of chicken after a workout. It seems logical that the protein gets digested, reaches the blood, and heads straight to your muscles. But not quite. A protein is a long chain of amino acids, folded into a complex three-dimensional structure, and its shape determines its properties. The protein in chicken, an egg, or beans was built by another organism for its own needs. Human cells can't just slot it in as a ready-made spare part. Besides, a whole molecule is usually too big to pass through the intestinal wall. So the body first takes the foreign structure apart. The first stage is in the stomach. The acidic environment changes the three-dimensional structure of proteins. They start to unfold, and this process is called denaturation. Picture a tightly tangled ball of yarn slowly coming undone. Hidden parts of the chain become more accessible. But the acid itself doesn't break protein down into amino acids. It creates the conditions for the enzyme pepsin. And pepsin works like molecular scissors and cuts the long chains into shorter fragments, peptides. Next, the partly disassembled protein moves into the small intestine. The acid gets neutralized, pepsin loses its activity, and the pancreatic proteases take over, the very ones that were stored switched off, so they wouldn't damage the place where they're made. Different enzymes attack different parts of the chain, and gradually free amino acids appear, along with short fragments of 2 or 3 amino acids. At the surface of the intestinal cells, single amino acids move in through special transporters, and dipeptides and tripeptides have a path of their own. But even these short fragments usually don't head into the blood hole. Inside the cell, enzymes more often cut them down to individual amino acids. Now the former structure is gone. For the cells, these are no longer amino acids from chicken, from an egg, or from beans, but universal building blocks. They exit the other side of the intestinal cell, enter the capillaries of the villus, and travel through the portal vein first of all to the liver. The liver uses some for its own tasks and regulates how they flow onward. After absorption, the amino acids enter a shared dynamic pool, a kind of distribution center, where the parts are constantly drawn out and used up. This isn't a warehouse of spare protein. The amino acids are in constant motion. From them, cells build enzymes, receptors, transport proteins, components of the immune system, skin, and the intestine. Some really does reach the muscles and goes into muscle protein synthesis. But the muscles don't get all of it. The intestine and liver take their share. The rest is distributed among the tissues. And if some amino acids aren't needed, the body can remove their nitrogen-containing group and send the rest into metabolism. So the protein you eat doesn't head straight to your muscles. First, it stops being the food's protein, gets taken apart into pieces. And only then does the body decide where to use them. But protein isn't the only thing the body first takes apart, then puts back together. Picture a drop of oil in a glass of water. No matter how you stir, after a while the oil gathers back at the surface. Fat barely dissolves in water, and inside the intestine it's a watery environment. And blood and lymph are mostly made of water. So how does fat get in? The body changes its form several times. First, the movements of the intestine physically break large fat droplets into many small ones, giving the enzymes access to a larger surface area. But small droplets tend to merge back together. Here bile salts help out. Each such molecule has one side drawn to fat and the other to water. They coat the droplets and keep them from merging. They don't cut the fat molecules. Their job is to keep many small droplets apart. Then pancreatic lipase joins in. Most dietary fat consists of triglycerides, like a backbone with 3 fatty acids attached. Lipase snips some of the bonds and turns them mainly into free fatty acids and monoglycerides. But even these products interact poorly with water and can't travel freely through the intestine. So bile salts create the next transport form, mixed micelles. A micelle isn't a sealed capsule, but a small dynamic structure, a kind of molecular ferry. It carries the fatty cargo to the wall of the intestine, but doesn't itself go into the cell. At the membrane, the fatty acids and monoglycerides leave the micelle and move inside, while the bile salts stay outside and get used again later. You'd think the fat could now go straight into the blood. But the cell does almost the opposite. It reassembles triglycerides from the pieces it received and packs them together with phospholipids, cholesterol, and proteins into a particle, a chylomicron. Why take apart, then build again? Large molecules were hard to move across the membrane. After disassembly, their pieces could cross the barrier, and now, from them, you can assemble a transport particle fit to travel through the body. But chylomicrons are too big for most capillaries of the villus, so they take a different path. In the center of the villus, there's a lymphatic vessel. The chylomicrons enter it, move through the lymphatic system to the thoracic duct, and only then does the lymph pour into a large vein, and the fat finally ends up in the blood. So most long-chain fat gets there not directly, but by a lymphatic detour. There's an exception. Short and medium-chain fatty acids interact better with water and can go into the blood more directly, toward the liver. Glucose, amino acids, and fat still have to cross the border between the contents of the intestine and the body's internal environment. And the intestine is not a sponge. Picture a glucose molecule right at the wall. Between it and the internal environment stands the intestinal epithelium, one cell layer thick. Until the molecule has crossed it, the body hasn't gotten it yet. The barrier's task seems almost impossible. Let water and nutrients through, but hold back bacteria and large structures. For this, the surface is organized into folds, villi, and tiny projections called microvilli. Together, they create an enormous contact area. But the larger the surface, the longer the border you have to control. Our glucose passes through a mobile layer of mucus. In the small intestine, it isn't a solid wall, or it would get in the way of absorption and reaches the membrane, where a transporter grabs it along with sodium. Now it's in the cell, but not yet in the blood. An intestinal cell has two different sides. On the intestinal side, transporters that take substances in. On the opposite side, systems that release them into the tissues. This creates a directed movement. Glucose enters on one side and exits on the other, into a capillary toward the liver. Such a path through the cell is called transcellular. And what about the bacterium nearby? There's no suitable transporter for it, and it's too big. It can't slip between the cells either. They're joined by tight junctions. But this isn't fixed cement. It's a regulated filter. It lets through water, ions, and some small substances, depending on their size, charge, and the state of the barrier. This is the paracellular path. The intestinal barrier works not because it's sealed shut, but because its permeability is selectively regulated. Normally, whole bacteria don't cross an intact epithelium, and beneath it the immune system is always on duty. It doesn't wait for a breakdown. It calmly guards the border and helps the body not react to ordinary food and a normal microbiota. But the barrier isn't invulnerable. With infection, inflammation, or cell damage, its permeability can change. And then substances that usually stay in the lumen cross the border. This doesn't mean every bit of bloating is caused by a leaky gut. Permeability is a regulated physiological property, and its disruption can't be diagnosed from a single nonspecific symptom. What to take away? The protein you eat doesn't head straight to your muscles. It gets broken down into amino acids, which are then distributed among the intestine, liver, muscles, and other tissues. Disrupted bile flow can worsen the digestion and absorption of fats. And a leaky gut can't be diagnosed from bloating or discomfort. So glucose, amino acids, and fat cross the barrier, each by its own route. But some food is out of the body's reach entirely. Fiber, for example, your enzymes can't break down. So why do the bacteria in the large intestine finish eating for you? That's in the season finale. Subscribe to BioCode Lab, where the complex becomes simple.